About two operons today. An operon is made up of a promoter, an operator, and the genes that are transcribed to make mRNA. The first operon I was talking about is the trip operon. The trip operon is an example of a repressible operon, meaning that the operon is always transcribing mRNA to translate into proteins. In this case, the tryptophan amino acid. The reason why the trip operon is always on is because the regulatory gene, which is a little bit upstream from the actual operon, codes for an inactive repressor. Now this inactive repressor is unable to bind to the operator, as you can see. And so there's nothing stopping the RNA from going on and transcribing the genes to make mRNA. Now trip operon is regulated by negative feedback. This means that when there is an excess of tryptophan will bind to the repressor, making it become active. And when it becomes active, the repressor will bind to the operator, and this serves as a roadblock for the RNA polymerase, and the RNA polymerase is no longer able to go on and transcribe the genes to make mRNA. This turns off the trip operon until the levels of tryptophan go down till the tryptophan comes away from the repressor. The repressor goes back to being inactive, and RNA polymerase is able to again go on to transcribe for mRNA. The second operon I'm talking about is the lac operon. The lac operon is an example of an inducible operon, meaning that in its natural state it's always off. This basically, and it's always off, that means the regulatory gene codes for an active repressor, and the active repressor binds to the operator, and once again serves as a roadblock for the RNA polymerase to go on and transcribe to make mRNA. In the presence of lactose and in the absence of glucose, allolactose will bind to the repressor, making it become inactive. In its inactive form, it will no longer bind to the operator, so there's nothing stopping the RNA polymerase from going on and transcribing to make mRNA. Now that is an example of negative feedback, but the lac operon also has a form of positive regulation, that in the form of CAMP and CAAP. So, in the absence of glucose, there are higher levels of something called CAMP, which is cyclic AMP. And when cyclic AMP will bind to the inactive form of CAP, making it become active. And when it becomes active, it will bind behind an RNA polymerase and will help the RNA polymerase bind stronger to the promoter, and it will promote the RNA polymerase's transcription of genes to make mRNA. When glucose levels rise again, the level of CAMP falls, meaning there's not enough CAMP to bind to the CAP, which causes the CAP to go back to being inactive and no longer there to help out the RNA polymerase bind to the promoter and transcribe the genes. In eukaryotes, a form of gene regulation can be seen in the process of RNA splicing. What happens when DNA is transcribed in a eukaryote, what is created is something called pre-mRNA. Pre-mRNA consists of a 5' cap, which is a modified guanine nucleotide that acts to protect the genetic material in the mRNA as it exits the nucleus to go into the cytosol to then be translated into proteins, and also serves as a signal for where the mRNA has to go as well. Along with pre-mRNA also has the poly-A tail, which is just a string of adenine nucleotides that act as a buffer in the cytosol to prevent the genetic material once again from being eaten away before it can be translated. And pre-mRNA also consists of exons and introns. Now, introns are actually non-coding segments of the mRNA, now are not necessary in the translation of proteins. So the introns must be removed before the mRNA is able to go out into the cytosol and then translate for proteins. And the removal of introns is done by a complex known as a spliceosome inside the nucleus. The spliceosome consists of small RNAs. These small RNAs will base pair to the intron nucleotide sequences acting as a marker of where the intron would then be cut out from the pre-mRNA and where the exons will be stitched back together. As you can see, the intron is removed and then the exons are joined together to form the final mRNA, which consists, once again, of a 5' cap, the coding sequence made of all exons, its number of introns, and a poly A tail. The pathway in which DNA causes a cellular response. So it begins when a signal comes into the cell, which then signals are chromatin inside a nucleus to unbind, so the DNA is open to be transcribed to become mRNA. Transcription occurs in the nucleus with the help of RNA polymerase, and this creates pre-mRNA. Now, this pre-mRNA still has non-coding segments in the mRNA known as introns. And so these introns need to be removed before the mRNA can be made into a functional protein. So the pre-mRNA undergoes something called RNA processing. In RNA processing, which occurs at the spliceosome, the introns are removed and a 5' cap and a 3' tail are added to the cell. A 5' cap and 3' tail allows the mRNA to be transported to the cytosol without fear of the mRNA being broken down into parts by digestive enzymes inside the cytosol. And so when the mRNA gets into the cytosol, it undergoes something called translation at the rRNA, where the mRNA codes for a polypeptide. After it has coded for the polypeptide, the mRNA is degraded back into its components so it can go back and make more mRNA. And moving back to the polypeptide is shaped into its active form, which makes an active protein, which then itself causes a cellular response. And then after that, the protein is again degraded into its subunits that can make more proteins as well.